Thank you to the organizers for giving me the opportunity to present our work. My lab is at Northwestern University in Chicago, and my lab is interested in the roles of chondroitin sulfate in development and disease. So this is going to be a bit more of a basic research talk, and that hopefully will lead to some therapeutic approaches down the road. And today I'm going to talk about some novel work we have done that identified chondroitin sulfates as regulators of cell fate decisions during embryonic stem cell differentiation. So stem cells usually always interact with the microenvironment in a stem cell niche to drive their differentiation and lineage determination. So the microenvironment includes stromal cells. The microenvironment also includes extracellular matrix proteins. And as I just said, we are interested in a glycosaminoglycan called chondroitin sulfate, which has been shown to be both part of the extracellular matrix as well as cell surface proteins on stromal cells as well as of some ES cells. And just to give you an idea what chondroitin sulfates look like, a little bit about the biosynthesis here. Usually chondroitin sulfates are attached to core proteins to form proteoglycans. And usually there are a large number of disaccharide units that are modified by sulfotransferase enzymes. So these enzymes add sulfate groups to different positions in that disaccharide unit. And I'm going to talk a fair amount about this specific chondroitin sulfate here. It's called CSE, and you can see it's double sulfated, and it has been shown, and I show as well, that it has very specific biological functions that make it actually a very interesting molecule. So CSE will come up a bit later. Are chondroitin sulfates of any interest in development and disease? We believe yes, and we have been shown before that if you knock out a chondroitin sulfotransferase that is important for adding sulfate to the C4 position, you get a perinatal lethality with very severe chondrocyte defects, skeletal abnormalities. In breast cancer, when we do orthotopic tumor models and digest chondroitin sulfate in those developing primary tumors, we get large secondary tumors and a large increase in lung metastases, indicating that the presence of chondroitin sulfate actually suppresses a metastatic phenotype in these orthotopic breast cancer models. So these data and others as well, of course, seem to support the notion that chondroitin sulfates do play a fairly important role in development and disease. And today I'm going to talk about the role of chondroitin sulfate in embryonic stem cell differentiation, and these are mouse embryonic stem cells. The model we are using, we basically differentiate embryonic stem cells as embryoid bodies on a flat culture system. So here's day 0, where we have a 4-day of suspension culture, so you get cystic embryoid bodies. Then we transfer those to tissue culture plates. You get a flat culture system, which makes it easier, first of all, for factors to access the cells. Secondly, also, it makes it easier to image the cells thereafter. And these are just different stages of these differentiation steps. Undifferentiated ES cells, day 4, day 6, day 12. You can see by day 12, the whole plate is basically covered by cells of various lineages. A methodology we're using throughout this talk, I'm going to talk about throughout this talk, is the digestion with a bacterial enzyme called chondroitinase ABC. What that does basically, it digests this intact chondroitin sulfate chain into disaccharides, leaving the core protein and this little linker region here. And that linker region actually is fairly important because antibodies we use recognize that. So you can only recognize chondroitin sulfate once you have digested this. First question, of course, we asked, if we want to see if chondroitin sulfate plays a role in ES cell differentiation, is it expressed during ES cell differentiation? And again, as I just said, we use an antibody that recognizes this linker region here. After you digest with chondroitinase ABC. If you have no chondroitinase ABC, you get no signal. If you digest with chondroitinase ABC, there seems to be no chondroitin sulfate expression in undifferentiated EF cells that still contain LIF in the media. If you withdraw LIF, you see an increase in chondroitin sulfate. And if you differentiate embryoid bodies, you see with time you get a fairly large increase in chondroitin sulfate concentration. So with differentiation, you get an increase in chondroitin sulfate expression. Does that do anything to differentiation? This is now looking at the S cells plus LIV minus LIV, and we look at pluripotency markers here, NANOG and OCT4. And what you can see, when you withdraw LIV, you get a drop in both NANOG and OCT4 because the cells now start differentiating. And what you can see here, if you also digest with chondroitinase ABC, you see increased differentiation, meaning in this case NANOG seems to be dropping significantly faster when you don't have chondroitinase ABC, if you don't have chondroitin sulfates here, indicating that chondroitin sulfates do seem to affect differentiation in one way or the other. And to explore this further, we went ahead and did this embryonic body culture system. So in this next set of experiments, what we did, we cultured the cells as embryonic bodies for 12 days, and from day 0 to day 12, we digested with chondroitinase ABC. We supplied new chondroitinase ABC enzyme every 2 days to make sure whatever chondroitin sulfates are expressed get digested and eliminated continuously. Macroscopically, well, actually microscopically, but not on the single cell level, there was not much of a difference. So minus and plus chondroitinase ABC, both at day 4 and day 12 shown here, there was no difference visually of these cultures. We then did a marker analysis by qPCR, and there were some interesting changes we observed. So this is basically a marker analysis. The first two for pluripotency, so OCT4 and NANOG, there was not much of a difference here. As expected, expression is higher in the beginning when cells are undifferentiated, goes down when they differentiate. That was the same plus or minus chondroitinase ABC. Ectoderm markers looked similar. Endoderm markers looked fairly similar. Early mesoderm, axial mesoderm, paraxial mesoderm looked very similar. Where we saw a difference was in cardiac markers. So transcription markers, early cardiac development transcription factors, GATA4 and NKX2.5. There was a distinct loss of these transcription factors after we digested chondroitin sulfates. And the same held true for MLC2V, a structural protein of cardiomyocytes. So it appears that digestion of chondroitin sulfate does affect the cardiac lineage fairly specifically during S cell differentiation. And we also did Western blot analysis to look for cardiac myosin heavy chain here, and that again shows the same thing. Digestion with chondroitinase ABC seems to lead to a reduction. This is the quantification here. Good. Now, then we wanted a biological readout for this marker analysis we just showed. And the good thing is when you digest cells as embryonic bodies, you get the formation of cardiac foci, meaning the cells actually start beating. So they form cardiac foci that actually have electrical activity, muscle that actually beat, and you can see that as you probably just did. And these are fairly easy to score under the microscope, and you can use them as a readout to how efficient is your cardiac differentiation in your culture system. And we then went ahead and did that, again using the chondroitinase ABC digestion. And what you can see here is that in a number of experiments, and in the back is the average, digestion with chondroitinase ABC always leads to a fairly significant reduction in cardiac foci formation, which is in cooperation with the marker analysis I just showed you, that there seems to be a reduction in cardiac markers. So that makes sense. These are just individual experiments where we showed that, where we followed cardiac foci, and in each case you can see no treatment, they go up, but with chondroitinase ABC they stay low down or don't go up as much to the level you observe in normal wild-type cultures. Good. So far I showed you that chondroitin sulfates do play a role in cardiac differentiation of embryonic stem cells. Next, we wanted to see what signaling pathways do chondroitin sulfates affect. How do they have this effect on cardiac differentiation? So what we did some Western blot analysis of various signaling pathways that have been shown to play a role in cardiac differentiation. And just to cut that short a little bit, BMP, TGF-beta, MAP kinase, different MAP kinase signaling pathways. Here's the Western blot, here's the quantification. Were not really affected by chondroitin sulfate digestion with chondroitinase ABC. However, what we did see that the Wnt beta-catenin, so the canonical beta-catenin pathway, was affected by chondroitinase ABC digestion, and it was affected in a way that it was upregulated. So if you look here at beta-catenin, which is an important intercellular mediator of Wnt, the canonical Wnt pathway, it goes up. And when you look at one of the Wnt receptors, LRP6, and specifically here phosphorylation of LRP6, that also goes up. We followed that up with a Wnt beta-catenin reporter assay. That's a top flash luciferase assay. And just looking at these two down here, ES cells minus LIF, as well as embryonic body state 10, chondroitinase ABC treatment goes up, supporting what we've seen here in the Western blot analysis. So Wnt signaling seems to be positively affected by digestion with chondroitinase ABC. This is just a quick summary of the Wnt pathway. We don't really need too much of this. Just to visualize what I just showed you, LRP6 is a co-receptor up here. I showed you that phosphorylation of LRP6 was upregulated when we digest with ABC. Beta-catenin is here as an intracellular mediator that in the presence of Wnt protein goes nuclear and activates Wnt transcriptional programs. So you just have an overview of what kind of the main molecules are here in this pathway. What's interesting, though, about the Wnt pathway, beta-catenin pathway, is that it has a biphasic role in a cardiac differentiation of ES cells. So it plays numerous roles in ES cell differentiation, but when you only look at cardiac differentiation, and this has been shown before, so these are data that were out before, for early cardiac progenitor cells, Wnt plays a positive role. So Wnt beta-catenin promotes cardiac differentiation. So at early stages of embryonic body differentiation, Wnt plays a positive role. Later on, Wnt plays a negative role. So from day 6 to day 12, Wnt beta-catenin blocks cardiac differentiation. So a biphasic role of Wnt in ES cell cardiac differentiation. Now, we wanted to bring this together. So we have the data I showed you that chondroitinase ABC affects cardiac differentiation. We know that it affects Wnt signaling, and we know that Wnt has a biphasic role in cardiac differentiation. So knowing this, we postulated if chondroitinase ABC affects cardiac differentiation mainly through activation of the Wnt pathway, we can say that chondroitinase ABC treatment at early stages should increase the number of cardiac foci because early Wnt beta-catenin promotes cardiac differentiation. It should be clear, but I thought about that probably more than you. So it should, if you treat early, it should go up. If you treat with chondroitinase ABC at later stages, cardiac foci formation should go down because of the biphasic role of Wnt beta-catenin. And that is exactly what we see. So if we now treat only at early stages and then withdraw the digestion, we see an increase of cardiac foci. If we treat only late, we see, and black bars here again, we see a strong decrease in cardiac differentiation. This strongly indicates that the role of chondroitin sulfate, the biphasic role of chondroitin sulfate to control cardiac differentiation is due to its effects on Wnt beta-catenin signaling. Okay. So far I showed you that elimination of endogenous chondroitin sulfate by chondroitinase ABC digestion leads to increased Wnt beta-catenin signaling and has a biphasic effect on cardiac differentiation of ES cells. So we wanted to go then further and ask, could a temporal treatment with exogenous chondroitin sulfates be utilized as a treatment to promote cardiac differentiation of ES cells through the repression of Wnt beta-catenin signaling? And we and others have shown before that this CSE I mentioned in the beginning, the double sulfated CSE, actually does repress Wnt beta-catenin signaling quite drastically. So this is a paper we had in 2012. These are fibroblasts now. When you treat them with normally a control condition medium or Wnt3a condition medium, so here you stimulate the Wnt pathway, the canonical pathway, you see that staining for beta-catenin, you see that beta-catenin goes nuclear, and you see this strong increase in nuclear beta-catenin here compared to the control. If you treat with a chondroitin sulfate that has no effect on Wnt beta-catenin signaling, you see the same holds true. It does not affect the nuclear localization of beta-catenin. However, if you treat with CSE, that is almost completely abolished. You can see here there's a tiny little bit of nuclear beta-catenin, but most of it is absent. And we analyzed that in much more detail in that paper. But here we wanted to go into ES cells or differentiating ES cells and wanted to ask, can CSE also be utilized in this system to reduce Wnt beta-catenin signaling? And this is immunofluorescence analysis looking again for beta-catenin localization by immunofluorescence in controls. And we only need really to look at the green panel here, which is beta-catenin. In the controls, you see diffuse staining that goes nuclear when you treat with Wnt. The addition of C4S, chondroitin 4 sulfate, has no effect. Beta-catenin still goes nuclear. When you treat with CSE, there's no nuclear beta-catenin anymore. So in this system as well, CSE seems to downregulate Wnt beta-catenin signaling quite drastically. Again, the top flash reporter assay shows the same thing. In control, you have good signaling. With CSE, the Wnt response is drastically downregulated. Now, all of this would not make sense if CSE would not be expressed in our embryonic bodies, but it is. So we used an antibody here recognizing specifically CSE at day 12 of differentiation. So when you have, this is the panel that's important, DAPI, and here is the CSE antibody. This is a control without primary antibody. This is a control when we treat with chondroitin ABC. You eliminate chondroitin sulfate, so there shouldn't be a signal anymore, and there is no signal. So CSE downregulates Wnt signaling and is also present at fairly high levels in our embryonic body cultures. Good. And then lastly, we then also wanted to see, can treatment with CSE, exogenous CSE, can also have this biphasic effect I showed you with the digestion of endogenous chondroitin sulfate. So just to summarize, show that again. So CSE blocks Wnt signaling, as I've showed you. So if we block Wnt signaling early on, we block this positive effect of Wnt. If we block Wnt signaling later on, we block the negative effect of Wnt on cardiomyocyte differentiation. And so again, treatment with CSE early on leads indeed to a reduction of cardiac foci formation, beating cardiac foci formation. Here's the average. Whereas treatment later on, from day 6 to day 12 of our differentiation protocol, leads to a strong increase in cardiac foci formation. This is recapitulated by MLC2V expression, a cardiac marker expression. Here's the control. C4S does not change anything if you treat early or late, but CSE, when you treat early, day 0 to day 4, you get a strong reduction in MLC2V expression. If you treat late, you get a strong increase in MLC2V expression, supporting what I showed you here, again, the biphasic role of chondroitin sulfates, directing ES cell differentiation into the cardiac lineage, depending what time point you actually treat. Good. This is the summary. I showed you that CSE can inhibit Wnt beta-catenin signaling in ES cells as well as other cells. If we treat with CSE, the consequence of that early Wnt inhibition is a loss of cardiac differentiation. If we treat at later stages, when Wnt has a negative effect, the consequence of late Wnt inhibition by CSE is to enhance cardiac differentiation. And I have one slide that has to do with therapeutic approaches, but again, it's a slide for the future or where we would like to go. Currently, there are many cardiac regeneration strategies being developed. Many of them deal with deriving cardiomyocytes in vitro through differentiation steps, for example, of embryonic stem cells, and then supplying those in vitro differentiated cells to an injury site, in this case, the heart, obviously. iPS cells, obviously, is a new thing that also is considered in this case. People also consider inductive signals that activate endogenous resident progenitor cells. So in this case, you do not supply cells, but you actually activate endogenous stem cells, progenitor cells, to regenerate heart muscle, basically, in this case. I showed you that treatment with CSE is a differentiation signal that could be utilized to differentiate stem cells into cardiomyocytes. What I might actually be more interested in is the modulation of endogenous chondroitin sulfate levels in the heart, or genes that modulate the sulfation balance, which might actually be even more interesting, to actually promote endogenous regeneration programs after a cardiac injury has occurred. And we are currently working on this here. With this, I want to thank you for your attention. The work has been done at Northwestern University, like we said. People in the lab, Robert Prince did a good amount of this work. He's now back in Mainz, University of Mainz in Germany, to do his medical training. Catherine Willis was a grad student in the lab. She's now in Memphis, Tennessee, at the St. Jude's Children's Hospital, doing a postdoc there. And I'm happy to take any questions you might have. Thank you.
In the video that you showed, there were two clusters of cells beating, but I think they were not synchronized. So how do you think it's possible to synchronize the beating of the cells? Because I imagine that could be a limiting factor for transplantation of these cells.
Yeah, the way we analyze these cardiac foci, if they're not synchronized, we count them as different cardiac foci. Usually a cardiac foci is a synchronized block of cells that beat unisono, so are synchronized. If we see two different foci that beat in different rhythms, we count them as two different cardiac foci. I don't know if that answers your question.
And when you want to transplant these cells, you just select one cluster of cells?
Oh, this was not about transplantation.
No,
this,
I know that,
yeah.
but the ultimate goal is the transplant.
The ultimate goal probably would be to get differentiation of your whole culture to a degree that you have a very high percentage of cardiomyocytes. And I would assume at some point then later you would probably FAC sort your cells to make sure you only get the cells you want. And FAC sorting probably would be the most prudent thing to do to make sure you have, you can really select for the cells that have all the markers you want for your transplantation.
They might have still all the markers, but not be synchronized, and not beating in a synchronized way.
But wouldn't the cardiac pacemaker help you to do that?
To select cells which should be transplanted, I would probably go more for the marker analysis because as you said, in vivo they will be exposed to external electrical stimuli, whereas here we are looking at spontaneous stimuli, which will be a different scenario. So I think the marker analysis would be more important, where you carefully select only the cells that have all the markers you want for the cells you transplant. This was only like a biological readout here.
I used to work a lot with hyaluronic acid, which has also been implicated in a lot of these pathways. I just wondered, I think that chondroitinase ABC sometimes has some cross-specificity for the two. And could you explain a bit more about how specific you think that this effect is?
No, that's a very good point. It has been shown that chondroitinase ABC can affect HA as well. Yeah. On the other hand, in HA research, hyaluronic acid research, these effects I described here have not been described. So for now, I would postulate that these are chondroitin sulfate-specific effect, with the caveat that indeed chondroitinase ABC treatment can affect HA as well. On the other hand, we have CSE treatment here, which is basically endogenous digestion versus exogenous CS treatment, which gave us complementary results indicating that HA probably is not part of this phenomenon, biological phenomenon we see here. But that's a good point, yeah.
Thank you.
Thank you.